In this video, we are going to be going over the second free response question from the 2015 AP Biology exam. Cellular respiration includes the metabolic pathways of glycolysis, the Krebs cycle, and the electron transport chain, as represented in the figures. In cellular respiration, carbohydrates and other metabolites are oxidized, and the resulting energy transfer reactions support the synthesis of ATP. Part A. Using the information above, describe one contribution of each of the following in ATP synthesis: catabolism of glucose and glycolysis and pyruvate oxidation, oxidation of intermediates in the Krebs cycle, and the formation of a proton gradient by the electron transport chain. So first we have the catabolism of glucose and pyruvate oxidation. So there's going to be a net 2 ATP per molecule of glucose that are going to be generated in this first part of the pathway called glycolysis that runs from glucose over here to pyruvate over here. And substrate level phosphorylation is essentially just a process where you have a phosphorylated substrate and one of these phosphate groups is cleaved and the energy is used to append the phosphate group to a molecule of ADP, which is just adenosine with 2 phosphate groups attached to it, and this is going to form ATP. So as I said earlier, there's going to be a net of 2 ATP produced per glucose. There's actually going to be 4 ATP per glucose that come out of this process, but in the early steps of glycolysis, you have to phosphorylate the 6-carbon sugar twice, which is called the investment phase. So that's why there is a net of 2 ATP produced per molecule of glucose. In addition, there are going to be 2 molecules of NAD plus reduced to NADH per molecule of glucose, and this carries the energy used to build the proton gradient in the electron transport chain. And finally, acetyl-CoA is the input to the Krebs cycle. It's the end product of pyruvate oxidation, as you can see here. In the Krebs cycle, there are going to be more molecules of FAD and NAD plus to produce more electron carriers for the electron transport chain, and 2 molecules of GTP per acetyl-CoA entering the cycle are going to be produced. And these can be an energy source in their own right, or they can be converted into ATP. Finally, the electron carriers are going to be oxidized, donating electrons to the ETC and leaving protons in the matrix. And as the electrons and at the same time, the electrons from those electron carriers are going to be going down this chain of various molecules embedded in the inner membrane of the mitochondria. And each time these electrons go through, these protein complexes are going to pump protons from the matrix into the intermembrane space, and this is against the concentration gradient. And so when you've got all these protons in the intermembrane space, they're going to want to go back into the matrix because they're going to want to go from the high concentrations to the low concentrations. So they're going to the proton gradient is going to be used to provide the electromotive force that powers ATP synthase, which is going to be bound in this inner membrane along with these 4 complexes. It's not shown in the picture. And this will be used to generate most of the ATP in aerobic respiration. Part B. Use each of the following observations to justify the claim that glycolysis first occurred in the common ancestor of all living organisms. Nearly all existing organisms perform glycolysis. And the reason for this is that glycolysis developed in the earliest of organisms, and it's been around since then in almost every organism. And this means that organisms that use glycolysis have some sort of selective advantage over those that don't. And that's why they've persisted and dominated against organisms that may have used a different metabolic pathway. Glycolysis occurs under anaerobic conditions, and the reason why this is true is that the atmosphere of early Earth did not have free oxygen. And that meant that early prokaryotes in Earth's ocean had to rely on anaerobic processes to metabolize their food and produce energy. Glycolysis occurs only in the cytosol, and the reason for this is that the earliest organisms were prokaryotes, and prokaryotes lack organelles, membrane-bound, that is, membrane-bound organelles. Part C. A researcher estimates that in a certain organism, the complete metabolism of glucose produces 30 molecules of ATP for each molecule of glucose. The energy released from the total oxidation of glucose under standard conditions is 686 kilocalories per mole. The energy released from the hydrolysis of ATP to ADP and inorganic phosphate under standard conditions is 7.3 kilocalories per mole. Calculate the amount of energy available from the hydrolysis of 30 moles of ATP. Calculate the efficiency of total ATP production from 1 mole of glucose in the organism. Describe what happens to the excess energy that is released from the metabolism of the glucose. So for the first question, we're just going to multiply 30 moles times 7.3 kilocalories per mole to get 219 kilocalories available from the hydrolysis of 30 molecules of ATP to 30 molecules of ADP. And since the complete metabolism of glucose produces 30 molecules of ATP, if this process is 100% efficient, it should be equal to the amount of energy stored in a mole of glucose. But this isn't true. The molecule, 1 mole of glucose stores 686 kilocalories, which is much greater than 219, which means that the process is not 100% efficient. Some of this energy is going to be lost and transformed into another form of energy. And to calculate the efficiency, we're just going to take the energy available in the output of the process divided by the theoretical energy that should be produced. And that's going to be 219 over 686 is equal to 0.319, which is 31.9% efficiency. And what happens to the rest of the energy? Well, it's just going to be lost as heat. That's why when you breathe on your hand, it's kind of warm. And also why your muscles produce most of the heat in your body, because they're using up a lot of energy and producing a lot of ATP. Part D. The enzymes of the Krebs cycle function in the cytosol of bacteria, but among eukaryotes, the enzymes function mostly in the mitochondria. Pose a scientific question that connects the subcellular location of the enzymes in the Krebs cycle to the evolution of eukaryotes. So, one of the questions you might ask is, if the enzymes function in the cytosol of bacteria, which is bound by a membrane and a cell wall, but among eukaryotes, the enzymes function mostly in the matrix of the mitochondria, which is bound by 2 membranes. What if the mitochondria were once free-living bacteria that used the Krebs cycle to produce ATP, and they entered an endosymbiotic relationship with eukaryotes? That is, they were living inside of them, and this was beneficial for both the prokaryote and the eukaryote hosts. And this is the basis of what is called the endosymbiotic theory. And there are several pieces of evidence for this, and one of them is that mitochondria have their own genome. It's circular like that of a prokaryote, and it encodes several genes, including those that code for proteins needed for the electron transport chain, although it's only a few of the many, many proteins in the ETC that there are. And mitochondria have their own ribosomes, and they also replicate through binary fission just like prokaryotes, and they replicate independently of the cell cycle of the entire cell. Thanks for watching this video. Please like and subscribe for some more content.